Welcome to the Deep Dive. We're here to cut through the static and deliver those clear insights you're looking for. Today, we're kicking things off with a story that honestly really sets the stage. Let's talk about Rebecca. She discovered a hidden health issue after, well, a really tough family event. Her mother had a heart attack at just 44,
Mm.
and it turned out her cholesterol was like 3 times what it should be. Plus, she had these lumps, xanthomas.
San Thomas.
Yeah.
They're cholesterol deposits.
Right, under the skin, around the eyes, all signs that cholesterol was building up everywhere. Then Rebecca gets tested, and her own cholesterol, 420 milligrams per deciliter since birth. It was familial hypercholesterolemia, FH.
Yeah, FH. It's a genetic condition where the body just, it can't clear cholesterol effectively from the blood. It builds up. It's a really striking example, unfortunately quite common too. Rebecca's situation, it's not just about her high numbers. It really shines a spotlight on this whole world inside us, the world of lipids.
Exactly, which most of us just think of as, fat in our diet. Maybe something in a cell diagram we saw once.
Right. But lipids are so much more. They're this huge essential family of biomolecules. We literally can't live without them.
And that's what we're diving into today. We want to get beyond just dietary fat. We're talking cell membranes, hormones, energy storage, the whole picture. We'll unpack what lipids actually are, how they function in your body, and why understanding them is so relevant, especially in health and life sciences. We're pulling together insights from various key sources to give you a clear handle on these vital concepts without, getting lost in the weeds. So let's start at the beginning. When we talk about lipids, what's the defining feature? What makes a lipid a lipid?
Well, the fundamental thing, the property that groups them all together, is how they react, or rather don't react, with water. Think oil and vinegar. They just don't mix. That's the core idea. Lipids dissolve in organic solvents, things like ether, but not in water. Even the name lipid comes from the Greek word lipos, which means fat or lard.
Ah, okay. So that insolubility in water explains why they're so good at forming barriers, like cell membranes. They keep the watery bits separate. Precisely.
Yeah.
And why they work as certain hormones traveling through the bloodstream. They're designed for non-watery roles, in a sense. And within this big family, there are different types. We'll touch on waxes, think protective coatings. Then triacylglycerols, that's your main energy storage fat. Glycerophospholipids and sphingolipids, absolutely key for cell structures. And finally, steroids. They look quite different with a unique fused ring structure. Interestingly, they can't be broken down by hydrolysis like most other lipids can.
Okay, interesting. So for many of these, the building blocks are fatty acids. What's the deal with them? What makes them fatty?
Fatty acids are pretty neat molecules. Basically, they're long chains of carbon atoms, usually unbranched, with a specific group at one end, a carboxylic acid group. Now, that acid group is a little bit attracted to water. It's hydrophilic. But the rest of the molecule, that long hydrocarbon tail, that's strongly hydrophobic. It repels water. So the whole molecule ends up being mostly insoluble. Most of the ones in nature have an even number of carbons, usually somewhere between 12 and 20. Like lauric acid, that's got 12 carbons.
So they're just long chains. How do we get different types? Is it just about how long the chain is?
Length is part of it, yeah. But the really critical difference is the type of bonds between the carbon atoms in that chain. We split them into two main groups, saturated and unsaturated. Saturated fatty acids, or SFAs, have only single bonds between the carbons. Think of them like straight, rigid sticks.
Straight sticks. No?
Yeah, and because they're straight, they can pack together really, really tightly side by side. This close packing means lots of intermolecular forces, dispersion forces, holding them together. It takes more energy to melt them. So they have higher melting points and are usually solid at room temperature. Stearic acid is a good example, melts way up at 69 Celsius. Think butter. Lard.
Got it. Solid at room temp because they pack tight. So unsaturated must be different because of double bonds.
Exactly, that's the key. Unsaturated fatty acids, UFAs, have one or more carbon-carbon double bonds. If it's just one double bond, we call it monounsaturated. Oleic acid, like in olive oil, is a classic example. If there are two or more, it's polyunsaturated, or a PUFA. Linoleic acid, arachidonic acid, those are PUFAs. And here's where it gets really interesting biologically. Almost all naturally occurring unsaturated fatty acids have their double bonds in a cis configuration.
Cis, right, and that causes a bend.
Yes, a distinct kink or bend in the chain. It's not straight anymore.
Ah, so that kink must stop them packing so tightly together.
You got it. That irregular bent shape prevents tight packing. It's like trying to stack bent sticks versus straight ones. Fewer points of contact means weaker forces between molecules, which means much lower melting points.
So that's why olive oil is liquid at room temperature. It's those kinks.
That's exactly it. This tiny bend in the molecule has huge real-world consequences, from cooking oil consistency to how our bodies handle these fats. It's a real aha moment when you grasp that. Now, trans fats, which you hear about, are different. Their double bonds are in a trans configuration, which makes the chain straighter, more like a saturated fat, allowing them to pack better. That's part of the health concern.
Okay, that makes sense. And some of these are essential, right? We have to eat them.
Precisely. Our bodies can make most fatty acids, but some specific polyunsaturated ones like linoleic acid and linolenic acid, and sometimes arachidonic is included, we just can't synthesize them efficiently. We have to get them from our diet. They're essential for health.
And they're not just building blocks. Fatty acids like arachidonic acid get converted into other important things, right? Like prostaglandins.
Yes, this is fascinating. Prostaglandins are derived from that 20-carbon PUFA, arachidonic acid. They act like local hormones, incredibly potent even in tiny amounts. They have really diverse effects. They can raise or lower blood pressure, stimulate muscle contractions, think labor induction, and importantly, they mediate inflammation and pain when tissues get injured.
Wow. So when I take an aspirin for a headache,
You're blocking the production of prostaglandins. Aspirin, ibuprofen, other NSAIDs, non-steroidal anti-inflammatory drugs, they work by inhibiting the enzymes that make these specific prostaglandins from arachidonic acid.
incredible connection. Okay, and then there's the whole omega-3 versus omega-6 thing. What's the difference and why does it matter?
It's all about where that first double bond appears in the chain, counting from the omega end, the methyl end. Omega-6 fatty acids, like linoleic acid, have the first double bond at the sixth carbon, common in many vegetable oils, historically high in Western diets. Omega-3s, like alpha-linolenic acid and the famous EPA and DHA you get from fish oil, have that first double bond at the third carbon.
And this relates to that Inuit paradox, doesn't it? High-fat diet, but low heart disease rates.
Exactly. Their traditional diet, very rich in fatty fish and marine mammals, is packed with omega-3s. Research on them helped reveal the specific benefits of omega-3s. While the general advice shifted towards unsaturated fats overall, the Inuit experience highlighted that omega-3s, in particular, help make blood platelets less sticky.
Less sticky platelets mean lower risk of blood clots.
That's the idea. Fewer clots means lower risk of heart attacks and strokes.
Yeah.
But like many things in biology, it's about balance. Too much omega-3 can actually increase bleeding time, so you don't want to go overboard.
Right, balance is key. Okay, so we've got the fatty acid building blocks. Let's talk about the bigger structures they form, like waxes. They seem pretty simple.
Waxes are relatively simple, yeah. They're basically esters formed from a long-chain fatty acid and a long-chain alcohol. Think two long, greasy chains linked together. Their main job is protection, usually creating a water repellent coating on plant leaves and fruits to prevent water loss, on animal skin, fur, feathers for waterproofing. We use them commercially too. Beeswax, carnauba wax for cars and furniture, jojoba in cosmetics, lanolin from wool grease for skin creams.
Okay, protective coatings, and the big one for energy storage, triacylglycerols, often just called triglycerides.
Right, these are absolutely crucial. Triacylglycerols are triesters, meaning three ester links connecting a glycerol molecule to three fatty acid molecules. Glycerol is a simple three-carbon alcohol. This is the main way animals store energy. Think about hibernating animals, like that polar bear example. They live off their stored body fat, their triacylglycerols, for months. It's incredibly efficient storage. Fat provides more than twice the energy per gram compared to carbohydrates or proteins.
And this brings us back to the simple fat versus oil distinction, based on whether they're solid or liquid.
Exactly. It comes down to the types of fatty acids they contain. Fats are triglycerides that are solid at room temperature. They usually come from animal sources and have a higher proportion of saturated fatty acids, giving them higher melting points. Butter, lard, beef tallow. Oils are liquid at room temperature, usually from plants, higher in unsaturated fatty acids with those kinks, so lower melting points. Olive oil, corn oil, canola oil.
Although you mentioned coconut and palm oil are exceptions. Plant-based, but solid.
That's right. They're unusual plant oils because they have a very high percentage of saturated fatty acids, especially lauric acid, 12 carbons. That makes them solid or semi-solid, even though they're from plants.
Interesting. Okay, so these triacylglycerols, these fats and oils, can be changed chemically. This matters for food processing and how our bodies use them, right?
Definitely. A major industrial process is hydrogenation. This is where hydrogen gas is added across the double bonds in unsaturated fatty acids, turning them into single bonds.
So you're essentially saturating them, making liquid oils more solid.
Exactly. That's how liquid vegetable oils are turned into semi-solid margarines or solid shortenings for baking. Manufacturers can control the extent of hydrogenation to get the texture they want.
But this is where trans fats come into the picture, isn't it?
Yes, this is the downside. During partial hydrogenation, where not all double bonds are saturated, some of the natural cis double bonds can get isomerized, flipped into the trans configuration. And as we mentioned, trans fatty acids have a straighter shape, more like saturated fats. The health concern is that they raise the bad LDL cholesterol and lower the good HDL cholesterol, increasing the risk of heart disease. So checking labels for partially hydrogenated oils is important, although some trans fats do occur naturally in small amounts in dairy and beef.
Good to know. So hydrogenation changes fats. How does our body break them down?
That process is called hydrolysis. Hydro meaning water, lysis meaning splitting. In digestion, enzymes called lipases, along with water, break the ester bonds in triacylglycerols. This releases the glycerol and the three fatty acids, which can then be absorbed and used by the body. Strong acids can also do this, but in our bodies, it's primarily enzymes.
Okay, and then there's saponification. You said that's soap making.
It literally is. Saponification is the process of heating a fat or oil with a strong base, like sodium hydroxide, NaOH, or potassium hydroxide, KOH. This breaks the ester bonds, but instead of free fatty acids, you get glycerol and the salt of the fatty acids. And those fatty acid salts, that's soap.
Wow, so fat plus lye equals soap. Simple chemistry with huge impact.
Absolutely. NaOH gives you hard soap bars. KOH tends to make softer liquid soaps. The type of fatty acids in the original fat also affects the soap's properties.
Amazing. Okay, let's shift gears to phospholipids. You called them the architects of our cells. What makes them architectural?
Their structure is perfectly suited for it. They're similar to triacylglycerols, but instead of three fatty acids, they have two. The third spot on the glycerol backbone is attached to a phosphate group, which is then linked to an amino alcohol, like choline or ethanolamine. This creates a molecule with two distinct parts: a charged polar head, the phosphate and amino alcohol part, that loves water hydrophilic, and two nonpolar tails, the fatty acid chains, that hate water hydrophobic.
So, a water-loving head and water-fearing tails. How does that build a cell membrane?
It's ingenious, really. When you put these molecules in water, they spontaneously arrange themselves to hide their hydrophobic tails from the water and expose their hydrophilic heads. The most stable arrangement is a lipid bilayer, two layers of phospholipids. The heads face outwards, towards the watery environment inside and outside the cell, and the tails face inwards, creating a nonpolar core in the middle.
So the membrane is basically this double layer acting as a barrier.
Exactly. It forms the fundamental structure of all cell membranes, defining the boundary of the cell and controlling what gets in and out.
Are there different kinds? And do they have health implications?
Yes. The main types based on glycerol are glycerophospholipids. Lecithins containing choline are abundant in brain and nerve tissue, also egg yolks. Cephalins contain ethanolamine or serine. Then there are sphingolipids like sphingomyelin. They use a different backbone molecule, sphingosine, instead of glycerol. Sphingomyelin is crucial for the myelin sheath insulating nerve cells.
Myelin sheath. That sounds familiar.
It is, and problems here have serious consequences. In multiple sclerosis, or MS, the immune system attacks and destroys the myelin sheath. Losing that sphingomyelin insulation disrupts nerve signal transmission, causing all sorts of neurological symptoms.
Wow.
Another critical example is in premature babies. Infant respiratory distress syndrome, IRDS. Their lungs haven't produced enough pulmonary surfactant, which is a mix primarily of phospholipids like lecithin and sphingomyelin. This surfactant is needed to reduce surface tension in the alveoli, the tiny air sacs. Without enough surfactant, the alveoli collapse, making breathing extremely difficult. Doctors actually measure the lecithin to sphingomyelin ratio, the LS ratio, in amniotic fluid to gauge fetal lung maturity.
It's incredible how vital these molecules are right from birth. Okay, let's move to the last major group. Steroids. Cholesterol gets all the press, but it's more complex than just bad, right?
Definitely. Steroids are structurally very different from other lipids. They all share a characteristic core structure: 4 fused carbon rings, 3 6-membered rings, and 1 5-membered ring. It's called the steroid nucleus. And cholesterol is perhaps the most well-known steroid. It's technically a sterol because it has a hydroxyl OH. It's incredibly important. It's a vital component of cell membranes, adding rigidity. It's abundant in the myelin sheath, brain tissue. And crucially, it's the starting material, the precursor for making vitamin D, all the steroid hormones, and bile salts used in digestion.
So our body makes it. We don't just get it from steak and eggs.
That's a key point. Your liver is constantly synthesizing cholesterol. We also absorb it from animal-based foods, meat, milk, eggs. Plants don't contain cholesterol. The recommendation is generally to limit dietary intake, like the AHA suggests, around 300 milligrams per day, because our bodies make what we need.
And this brings us back full circle to Rebecca. Excess cholesterol causes problems.
Exactly. When there's too much cholesterol, particularly carried by certain lipoproteins, it can accumulate in artery walls, forming plaque. This is atherosclerosis. The plaque hardens and narrows the arteries, restricting blood flow and increasing the risk of heart attacks and strokes dramatically. That's why someone like Rebecca needs careful management, looking at her whole lipid profile, total cholesterol, HDL, LDL, and using diet, exercise, and often medications like statins to control it.
Okay, let's clarify that LDL versus HDL thing. Bad versus good cholesterol. What are they really?
Right, so cholesterol and other fats can't just dissolve in the blood. They need to transport. That's where lipoproteins come in. Think of them as transport spheres. They have an outer layer of proteins and phospholipids, which, like water, and they carry a core of nonpolar lipids, like triacylglycerols and cholesterol esters on the inside. LDL stands for low-density lipoprotein. It's called bad because its job is to deliver cholesterol to the cells and tissues. If you have too much LDL, or if it's not cleared properly, it ends up depositing cholesterol in artery walls, contributing to plaque.
Okay, LDL delivers it, so HDL must.
HDL is high-density lipoprotein. It's the good guy because it does the opposite. It acts like a scavenger, picking up excess cholesterol from the tissues and arteries and transporting it back to the liver. The liver can then process it for removal from the body, often by converting it into bile salts. So high HDL levels are generally protective against heart disease.
Got it. LDL delivers, HDL removes, and cholesterol is also the basis for hormones.
Yes, steroid hormones. These are powerful chemical messengers derived from cholesterol that regulate a huge range of functions. You have the sex hormones, testosterone, and others in males, estrogens and progesterone, in females controlling sexual development, reproduction, etc. Synthetic versions are used in birth control pills. Then there are the adrenal corticosteroids from the adrenal glands. Cortisol manages stress response and metabolism. Aldosterone regulates salt and water balance. Synthetic versions like prednisone are potent anti-inflammatories.
And what about anabolic steroids?
Those are synthetic versions or derivatives of testosterone. Some athletes misuse them to build muscle mass, but they come with really severe health risks. Liver damage, heart problems, hormonal imbalances, psychological effects. Definitely not something to mess with.
It's amazing how that one core steroid structure leads to so many different powerful molecules. Okay, finally, let's loop back to cell membranes, tying it all together.
Right. Cell membranes are the gatekeepers. These essential barriers separate the inside of the cell from the outside world, controlling everything that goes in or out. And their fundamental structure, as we said, is that lipid bilayer made of phospholipids. Hydrophilic heads out, hydrophobic tails in, creating that selective barrier.
But you also said it's not just a static wall, it's fluid.
Exactly. That's the fluid mosaic model. Remember those kinks in the unsaturated fatty acid tails of the phospholipids?
Yeah, they prevent tight packing.
Right, so the phospholipids aren't locked rigidly in place. They can move around laterally, making the membrane fluid and dynamic. And it's a mosaic because embedded within or attached to this fluid lipid layer are various proteins. Some sit on the surface, others span the entire membrane. Cholesterol is also dotted throughout, adding some stability and regulating fluidity. Plus, there are carbohydrate chains attached to proteins and lipids on the outer surface, important for cell recognition and communication.
So how do things actually cross this fluid selective barrier?
Several ways. Small, nonpolar molecules like oxygen and carbon dioxide can just slip right through by diffusion, moving from high concentration to low concentration. Simple passive transport. Water can also move across, although slower. Some ions and larger polar molecules need help. Facilitated transport uses specific protein channels or carriers embedded in the membrane. It's still passive, following the concentration gradient, but the protein helps the substance across faster. Glucose often moves this way.
And what if a cell needs to move something against the gradient, like pumping ions?
That requires energy. It's called active transport. The cell uses energy, usually from ATP, to power protein pumps that move substances from low concentration to high concentration, the opposite of diffusion. This is crucial for maintaining ion gradients, like pumping sodium out and potassium into cells, which is essential for nerve function and many other processes. Ions can't just diffuse across the lipid core because of their charge.
Wow. So from Rebecca's high cholesterol through fats, oils, waxes, soaps, hormones, right down to the fluid dynamic barrier of every single cell, lipids are just incredibly fundamental.
They really are. We've covered a lot, from energy storage in triglycerides to the essential structure of phospholipids in membranes, the signaling power of steroid hormones, and even how fatty acid shapes determine physical properties. Their inability to mix with water is the key to almost all of their diverse and vital roles.
So, for everyone listening, maybe the next time you think about fats in your diet or just how your body works, take a moment. Consider the unseen constant work of these lipid molecules. They're shaping your health, your energy, the very definition of your cells. Makes you wonder, doesn't it? What other complex biological functions hinge on the subtle chemistry and shape of molecules we might otherwise overlook? Something to ponder.
We really hope this deep dive has helped clarify these crucial concepts, providing some aha moments and making the world of lipids a bit less mysterious and more practically understood. Thanks for joining us.